everyone, welcome back to Biophilia's video on part 3 of sexual reproduction in flowering plants. So before starting this video, do like, share and subscribe and hit the bell icon so that you get notified for the future videos. So do you remember that in the previous videos I had told you that the male pollen grain is going to be immotile, and because of it is immotile it needs some medium so that it can reach to the female reproductive part. So here we are going to understand the mechanism how the male is going to reach till the female. So the process is going to be called as pollination. So how would you define pollination? The transfer of pollen grains from the male part to the stigma will be called as pollination. A pollination जो होता है, that is because the male, that is the pollen grains, and the female, that is the embryo sac, these are going to be non-motile. Now since they are non-motile, they need some external mediums so that they can come together for fertilization. So pollination is going to be of different types. So types of pollination there are three. First is going to be autogamy. Autogamy क्या होता है? Wherein the transfer of the pollen grain is going to be from the anther to the stigma of the same flower. Okay, so here you have to remember that is transfer is on the same flower. Second type of pollination can be geitonogamy. Geitonogamy is nothing but transfer of pollen grains, that is from anther to stigma of another flower but of the same plant. Now here what was happening in autogamy, same flower tha, so ultimately that is going to be the same plant. Now here the flower has been changed, but that another flower is also going to be present on the same plant. Third type of pollination होता है, that is xenogamy. Xenogamy में anther से stigma का transfer होगा, but in this case the transfer would be from another flower to different plant of the same species. अब ये सब में जो था that you have to remember कि autogamy और geitonogamy में since the plant is same where the pollen is reaching and from where it has left, because of which autogamy or geitonogamy में there is going to be same genetic material or similar genetic material in both pollen grains as well as the female part of the flower. जबकि xenogamy में क्या होता है, because जो pollen grains का source है and the female gamete, they both are going to come from different plants, and since different plants, that means there is going to be different sources of genes. Now when genes are going to differ in the gametes, there is going to be variations occurring. So xenogamy is going to be a much better part of pollination. So in the figure you can understand A, that is autogamy, that is occurring on the same flower. B is the geitonogamy, that is same plant but different flower, whereas C, that is your xenogamy, same species, different plant and different flowers. Now let's understand some adaptations that plant undergo for ensuring that it undergoes self-pollination. So self-pollination होने के लिए first adaptation can be that is bisexuality. That means both the essential whorls. कौन से essential whorls थे? First was the male part, that was androecium, and second was the female part, that was gynoecium. So when these both whorls are going to be on the same flower, that is bisexuality. अब same flower पे है, so the plant का जो pollination होगा that would be autogamy and that would ensure self pollination. Second हो सकता है homogamy. Homogamy यानी क्या होता है? When the maturation of androecium and gynoecium is occurring at the same time. जब maturity same time पे होगा, so there is going to be synchrony in the release of the gametes. अब gametes अगर एक ही time पे release होंगे या develop होंगे, that is going to ensure self pollination. Third होता है cleistogamy. Cleistogamy is a condition wherein the flower doesn't open, so it was always going to remain in its bud state. अब bud state में रहेगा, so what is going to happen? The anthers and the stigma are going to lie very close to each other. Now when they are lying very close to each other, the pollen grains are going to come in contact with the stigma, and this contact is nothing but going to ensure self pollination. So there are some plants wherein cleistogamy condition can be seen, that is Viola, oxalis, camelina. These are the flowers wherein cleistogamy is seen. अब camelina flower जो होता है, the same plant can either have open flowers, तो open flowers condition को बोलते हैं chasmogamous flowers, whereas the flowers are closed, then it would be cleistogamous flowers. So camelina is a plant wherein it is going to produce both types of flowers, that is open as well as closed flowers. Now let's see some advantages of cleistogamy. Cleistogamy, bud pollination, why is it still occurring in these plants? So advantages में होता है कि it is going to ensure seed formation even if there is absence of any sort of pollinating agent. Suppose pollination नहीं हुआ but still in the bud state, because of close proximity, still the seed formation is going to occur. Second advantage है that it would be much cheaper for the plant. Cheaper in sense because plant doesn't have to use its energy for nectar or fragrance sources, because those plants are using pollinating agents. They need to provide fragrance or nectar to attract some pollinating agents. तो अगर bud pollination हो रहा है, that is if the flowers are cleistogamous, then what is going to happen, the plant doesn't need to produce any sort of nectar. तो nectar बनाने के process का जो energy होगा, that would be saved and that would be a helpful or a benefit for the plant. But all these were the advantages, but still there is one disadvantage of cleistogamy condition, that is the offspring that is going to be produced because of this sort of pollination, there would be limited genetic diversity. Limited genetic diversity क्यों है? Because the gametes are going to come from the same source. अगर same source से gametes आ रहे हैं, then what is going to happen? The genetic material is going to be a more similar case. Similar case की वजह से there is going to be limited genetic diversity. Now let's see fourth condition, that is bud pollination. Bud pollination is again similar type of cleistogamy wherein the flowers do not open, they remain in the bud stage and pollination occurs before the opening of the flowers. So examples can be pea and rice wherein bud pollination can be seen. Now let's understand some agents of pollination. तो agents of pollination कौन से type of pollination के अंडर आएंगे? They can either come under chitinogamy or xenogamy. So agents of pollination दो हो सकते हैं, that can be abiotic agents including wind and water, or biotic agents majorly animals. So biotic agents से जो pollination होता है, that is going to be in majority of the plants, whereas abiotic agents का जो pollination होता है, that is going to be used by only few plants. Now here can be seen the agents of pollination that were birds or insects. You can see those in the photographs. Now let's understand all these agents in detail one by one. So first let's understand the wind pollination, and wind pollination is also called as anemophily. Anemophily is a more common type amongst the abiotic pollination. Abiotic में कौन से दो आ रहे थे? Wind and water, out of which wind is going to be a more popular or more common type of pollination. So these flowers, those who are undergoing wind pollination, there has to be some characteristics for this type of pollination. So first characteristics होता है, the pollen grains are going to be lightweight and non-sticky. Now why this is going to happen? Because if the pollen grains are heavy, they will not be lifted by the air. Okay, and even if they are sticky, the wind blow hogi but they will not come out of the anther. So it is very important that the pollen grains are light and non-sticky so that they can be easily carried away by the air currents. Secondly, whatever flowers are there, they have to have their stamens well exposed. Stamens अगर well exposed नहीं होंगे तो क्या होगा, wind आएगी but the pollen grains would not be carried away. So there should be well exposed stamens so that the pollens are easily dispersed in the wind currents. Third point आता है flowers, that is the female flowers, they should have large feathery stigma. लार्ज फेदर स्टिग्मा से क्या होगा, जो विंड ब्लो की वजह से पोलन जो कैरी हुए थे दे हैव टू बी ट्रैप्ड बाय द फीमेल पार्ट ऑफ द फ्लावर, दैट इज द स्टिग्मा. So that should be a very large part so that more amount or more chances can come that the pollen grain comes and stick on the stigma. So flowers should have large feathery stigma. अब यहाँ पे एक important characteristic है, that is wind pollinated flowers will not have nectarines, मतलब these flowers are not going to produce any sort of nectar. One more important thing, that is presence of single ovule. Single ovule in every ovary, यानी जितने pollen grains आएंगे they have a better chance to fertilize a single ovule. Single ovule होगा, many pollen grains would be coming and landing on that. That would give a higher chance for fertilization and proceeding the further processes. So these wind pollinated flowers need to have single ovule. Secondly, over here these flowers need to be packed in inflorescence. Inflorescence से क्या होगा, all the flowers would be at single place, and because of the single place what would happen, there is going to be higher chances of fertilization. क्योंकि अगर वो scattered होंगे all over the plant, it will become difficult for the pollen grains to come and reach on all the flowers. So inflorescence से क्या होगा, the probability of reaching pollen grains is going to increase. So these wind pollinated flowers are going to be from the maize, wheat, sugarcane and bamboo. So here in the photographs you can understand that how these inflorescent or feathery stigmas will be present in these sort of wind pollinated flowers. Now let's understand about the water pollination, that is also called as hydrophily. Hydrophily is a pollination type which is going to be a rare condition in the flowering plants. So generally ऐसा देखा जाता है that only 30 genera out of these 30 they are only monocots. Only those prefer water pollination. So let's understand now what are the types or what is the characteristics of hydrophily flowers, यानी जो flowers water pollination prefer करते हैं, what are the characteristic of these flowers. So first of all, the pollen grains should be light and unwettable. अब ऐसा क्यों होना चाहिए, because again the water currents have to carry these pollen grains. अगर वो heavy होंगे they will sink into the water bodies. So they should be lightweight and unwettable, because if they get wet they may get damaged. So the unwettable property is going to be provided by the mucilaginous covering which would be over the pollen grains. Second आता है, there should be long sticky unwettable stigma. Again these plants are going to be inside the water, those who are going to undergo water pollination. So the stigma should also be long so that it can get and trap some pollen grains. It should be sticky so that whatever pollen grain comes, it should quickly just stick onto the pollen grain. And again it should be unwettable, because wetting of the stigma may rot it and may get destroyed. So unwettable conditions should be there also for the stigma. अब हमें यहाँ पे देखने हैं that pollination that is occurring by water, that can be classified into two categories, whether the fertilization is occurring at the surface of the water or it is below the surface of the water. So अगर surface of the water पे हो रहा है then you would call it epihydrophily, right? तो epihydrophily के बारे में let's understand some points. So here the female flower for the epihydrophily is going to have a very long pedicel. That is, the female flower is going to be tall enough that it is going to reach the surface of the water. अब मेल फ्लावर्स जो होते हैं they are going to release the pollen grains onto the surface of the water. So surface of the water पे पहुंच गए हैं pollen grains and the female flower is ready to trap these pollen grains on the surface of the water. So when the pollen grains are going to be carried passively by the water currents, they are going to eventually reach the female flowers and then the fertilization is going to occur. अब ये surface of the water पे हो रहा है, so this is going to be epihydrophily and this type of pollination is seen in Vallisneria. Second type होता है that is hypohydrophily. Hypo, that is below, so this is going to occur below the surface of water. So since it is occurring below the surface of water, so the female flowers are going to remain submerged in the water. Female flowers पानी के नीचे होंगे, whereas the pollen grains would be released onto the surface of the water or just below that, and they would have long ribbon-like structure that would help them to be move in the water. और जैसे ही वो move करेंगे into the water, somewhere they are going to reach the stigma and then achieve the pollination. So hypohydrophily can be observed in Zostera and Zostera is a seagrass, right? So Zostera में होगा hypohydrophily. Now here you have to remember that majority of the aquatic plants that can be seen, they are going to have their flowers immersed above the level of the water. यानी उनके सारे flowers पानी के surface के ऊपर दिखेंगे. Now those flowers whose complete structure is going to be seen out of the water, majorly they are going to be pollinated either by insects or by wind. So जिनके flowers पानी के surface के ऊपर है, they are not pollinated by water. Only those who are below the surface of water, they are pollinated by water. So यहाँ पे कौन से aquatic plants है whose pollination occurs by wind or insects? That is water hyacinth and water lily. So you can see in the diagram adjacent, water lily के flowers पानी के ऊपर है. Those who are pollinated by insects, as well as water hyacinth, that is Eichhornia, that is also called as terror of Bengal, right? So again this is going to be pollinated by insects. Now let's understand about insect pollination. Insect pollination को entomophily भी बोलते हैं and it is going to be the most common biotic agent. That is, insect is going to be the most common biotic agent for pollination. So generally insects में से भी bees or the honey bees are going to be the most common insect as the pollinating agents, but there are some other insects also, those who can act as pollinators like butterflies, flies, beetles, wasps, ants, moths. All these can also help in pollination. Now there are some flowers, those who prefer to get pollinated by insects. They are as yucca, salvia, ficus, calotropis, centuria, alistoria. All these are going to be the flowers who prefer insect pollination. Now out of this you have to remember that bombax and callistemon, this is flower that is going to undergo bird pollination. So bird is going to help in the pollination of these flowers. Now these two plants are going to be pollinated by bats, right? And this cobra plant is going to be pollinated by snails. So snails, as you know, these are the very slow creatures. So this is the flower that is pollinated by snails. And last two, that is Santalum and Mischlia, these are the flowers that are going to be pollinated by snakes. Right, so this was some extra information that other than insects there are some other creatures that are going to help in pollination. So now let's understand what are going to be the features for the flowers that are going to be pollinated by insects. So first characteristic आता है, that is majority of the insect pollinated flowers are going to be large size. Now reason for large size, that they can attract from a very large distance for these pollinators to come and pick up the pollen grains. So they need to be a very large size for the visibility purpose. Second होता है, if the flowers of the plant are going to be small sized, then they are going to be collected and clustered into a inflorescence. Inflorescence में अगर सारे small flowers come together, क्या होगा, inflorescence will become bigger in size, and this can also be helped for visibility purposes. Now again, for visibility from a very long distance, and for the feeling that the pollinator should come, so the flowers are going to be colorful and fragrant. So also because of the fragrance, the pollinators can be attracted towards the flower. So for the fragrance purposes, the flowers should have nectarines. Nectarines क्या होते हैं? Those who are going to produce nectar, and because of this nectar only the pollinator is going to come and get attracted. So here secondly, sticky pollen grains होने चाहिए. Sticky pollen grain से क्या होगा, the pollinator is going to come onto the flower or visit the flower, उसके sticky nature की वजह से it is going to stick all over the insect. So once it sticks onto the insect, it can be easily transported from one flower to the another flower with the pollinator. लास्ट होता है कि some flowers are releasing fall odor, that is they don't smell pleasant. So fall odor flowers are majorly pollinated by flies and beetles, but generally majority of the flowers are producing sweet nectars that is giving a very good fragrance. Now here you have to understand one topic, that is floral rewards for the insects. अब floral rewards क्या होते हैं? Plant understands that if the pollen needs to be pollinated, there should be some rewards for the insects so that they get attracted for the pollination. तो plant क्या क्या देता है as floral rewards? First, it can either give nectar, that is sweet juice produced by the flowers. So nectar is going to be collected by the honeybees, जिसकी वजह से by mistake there is going to, by unknown methods there is going to be pollination occurring. Second can be pollen that can also be provided by the insects. So some pollen can be kept by the insects and rest of them can be used for pollination. Third होता है to provide a safe place to lay eggs. So amorphous जो flower है, amorphophilous, this flower is going to provide a place to lay eggs. तो जैसे ही insect आएगा to lay eggs, that is going to pollinate the pollen grains to the another flower. Fourth आता है again to provide place to lay its egg. So example, a very classic example of pronuba moth that is going to lay eggs in the ovary of yucca plant. So yucca plant के अंदर जब lay eggs होगा, so when the larvas get into the moth, so they will come out of the flower. So when they come out of the flower, they are going to take the pollen grains along with them, और उसकी वजह से the yucca plant can get pollinated. अब एक टॉपिक आता है that is pollen और nectar robbers. अब कई बार क्या होता है, some insects instead of pollinating, what they do, they just consume the pollen or the nectar that is provided as floral rewards. वो बिना pollination के ही they go away from the tract and they consume that pollen or nectar. So if pollen or nectar is being consumed without bringing pollination, then they would be called as pollen or nectar robbers. So pollen or nectar robbers का title किसको दिया जाता है? Generally the honeybees. They are going to collect the nectar and pollen and they will store it in their beehives, and when they will store this is going to convert into honey that we use. So these are the pollen or the nectar robbers as honeybees. Now let's understand some outbreeding devices. अब outbreeding devices क्या होते हैं? These are going to be the features or the devices that are going to be introduced in the plants so that they do not undergo or they discourage self-pollination. So starting में हमने देखा था कि there were some factors that were responsible for self-pollination, but outbreeding devices होते हैं that they are going to encourage cross-pollination. और क्रॉस पॉलिनेशन क्यों होना चाहिए? सो दैट देयर इज नो लॉस इन जेनेटिक वेरिएशन और इन ब्रीडिंग डिप्रेशन से दे कैन ओवरकम. बिकॉज ऑफ दैट cross pollination is very important. So let's see some outbreeding devices one by one. So पहला outbreeding device है that is unisexuality or also called as dicliny. So production of unisexual flowers is going to ensure that there is cross pollination. So यूनिसेक्सुअल फ्लावर्स प्रोड्यूस होने के लिए देयर कैन बी टू कंडीशंस. पहला होता है मोनोशियस प्लांट्स एंड सेकेंड इज डायोशियस प्लांट्स. सो मोनोशियस प्लांट्स में क्या होता है? मेल एंड फीमेल इज गोइंग टू बी प्रोड्यूस्ड ऑन द सेम प्लांट. Okay, so female or male parts are not going to be on the same flower, but they would be on the same plant. Now because of monoecious plants, that is male and female on the same plant, there is going to be prevention for autogamy, यानी सेल्फ फर्टिलाइजेशन नहीं होगा, बट जिटनोगैमी इज नॉट प्रिवेंटेड. जिटनोगैमी क्या था? पॉलिनेशन ऑकरिंग फ्रॉम द डिफरेंट फ्लावर्स ऑफ द सेम प्लांट. सो यहाँ पे मोनोशियस प्लांट में क्या होता है? ऑटोगैमी इज प्रिवेंटेड बट जिटनोगैमी इज नॉट prevented. So here you can remember the examples, that is maize, castor and coconut. Then dioecious plants में क्या होता है? Male and female flowers are going to be produced on two separate plants. अब two separate plants आ गए यानी genetic composition is going to differ. So that is going to prevent autogamy as well as geitonogamy. So question can come to you कि which plants have a condition wherein autogamy and geitonogamy is prevented. So examples are going to be Vallisneria, papaya and date palm. Now majority of the questions they like to confuse between coconut and date palm. Okay, the plants look very similar to each other, so they like to confuse between coconut and date palm ki who is going to prevent geitonogamy and who cannot. So coconut is going to be a monoecious plant. So monoecious plant है यानी क्या है? It is not going to prevent geitonogamy. So you can remember it as CM, Chief Minister, that is CM. Coconut is going to be monoecious. जबकि date palm क्या होगा? Date palm, that is DD. Date palm is going to be dioecious, that is it is going to prevent geitonogamy. Now let's see the second feature for cross fertilization, that is डिक्नोगैमी. डिक्नोगैमी में क्या होता है? देयर इज गोइंग टू बी डिफरेंट टाइम फॉर मैच्योरेशन. अब मैच्योरेशन टाइम डिफरेंट होगा इवन इफ द पार्ट्स आर ऑन द सेम प्लांट. क्या होगा? अगर पोलन ग्रेन्स रिलीज भी हो जाए देन दे आर नॉट गोइंग टू बी एक्सेप्टेड बाय द सेम फीमेल पार्ट ऑफ द फ्लावर क्योंकि इट हैज नॉट मैच्योर्ड. और अगर मेच्योरिटी नहीं होगी in any one of the part, then that is not going to help in fertilization. So either if the pollen is released before the stigma becomes receptive, तो उसको बोलते हैं proto-androus condition, that can be seen in sunflower or in cotton. So proto-androus, proto यानी पहले, androus is the male part. So अगर male part पहले mature हो जाता है then that would be the proto-androus condition. For example, if the stigma, that is the female part, becomes more receptive, that is if it becomes more mature before the release of pollen grains, then that would be called as proto-gynous condition. Again, proto is first, gynous is the female part. So female part अगर पहले mature हो जाए, that is in case of ficus and allistonia, then that would be the प्रोटोगाइनस कंडीशन. थर्ड आता है सेल्फ इनकम्पैटिबिलिटी. अगर ऐसे कोई कंडीशन हो जाए कि pollen grains coming from the same plant, okay, pollen grains coming from the same plant or from the same flower, then the fertilization of the ovule is going to be inhibited. अगर ऐसा arrangement हो जाए then it is going to be called as self incompatibility. फोर्थ होता है कि in some plants the plant arranges its stigma and anther on different levels. अगर different positions, different heights पे अगर दोनों को arrange किया जाए, so it becomes difficult for the pollen grain to come in contact of the same stigma. अगर same stigma पे अगर pollen grains land नहीं होंगे, then what will happen? Self-pollination would be prevented. So that is what the plant wanted, to avoid self-pollination. So self-pollination को avoid करने के लिए these were the 4 techniques that can be used by the plant for encouraging cross-pollination. Now let's understand कि अभी तक जो हमने देखा था that was all regarding pollination, कि pollen आके stigma पे land कैसे करेगा. So once the pollen has landed on the stigma, what is going to happen next will be studied in pollen-pistil interaction. So pollen grains जो आते हैं, that is by the process of pollination. So pollination doesn't give you a guarantee कि the perfect right type of pollen grain is going to land on the stigma. It can be a possibility कि जो pollen land हुआ है on the stigma, that can be of the wrong type. If it is of the wrong type or coming from another species, तो fertilization नहीं होगा. Or else if it is coming from the same plant, and if the plant is self incompatible, that is it is not accepting the pollen grains from the same plant, so these pollen grains will not be utilized, they would go waste away. So now let's imagine for our study the pistil, that is the stigma, has received the correct type of pollen grain. So how does it understand कि ये जो pollen grain land हुआ है, it is going to be the correct time. So for the recognition of the pollen grain, whether it is the correct type or the wrong type, that is if it is compatible or incompatible, for that it is going to undergo a continuous chemical dialogue. And with the various type of chemicals, there is going to be a communication between the pollen grain and the pistil. Because of this reason, once the stigma understands that this pollen grain is compatible and can get fertilized, then what is going to happen? Post fertilization events are going to start. So first post fertilization events में जो series होती है, let's understand them one by one. So as soon as the pollen grain is going to reach and it is declared that it is compatible, the pollen grain is going to germinate. अब germinate हो रहा है, what does it mean? It is going to produce a pollen tube. A pollen tube बनेगा from one of the germ pores. Now this germ pores is nothing but the area wherein sporopollenin was absent. तो sporopollenin जहाँ पे missing है, from that the intine is going to form the pollen tube, and when this pollen tube is going to be formed, the pollen grain contents जो थे अंदर के, that is the male gametes and the vegetative cell, the cytoplasm, all that is going to flow into the pollen tube. So pollen tube जो होता है, it will proceed all over till the style and it will undergo to reach the ovary. अब जैसे ही ovary के पास it is going to reach, what is going to happen? The generative cell is going to divide and produce 2 male gametes. If it was released in 2 cell stage, और अगर 3 cell stage में ही release हुआ था, then what is going to happen? Directly the pollen grain is going to carry the 2 male gametes from the beginning. अब pollen grain के अंदर की ट्यूब बन चुकी है and the male gametes have been released into the tube. अब ट्यूब को पहुंचना है till the ovule. Now how is the pollen tube going to enter the ovule? That can be condition for 3. अगर ovule के अंदर entry is occurring from the micropyle से अगर entry हो रही है, then that would be called as porogamy. अगर चलाजा से एंट्री हो रही है then that is called as chalazogamy, and if it is entering from integuments then it would be called as mesogamy. So here down in the figure you can understand that how porogamy, then chalazogamy and mesogamy occurs when the pollen tube is going to enter into the ovule. So entry of ovule is going to have three types. Next point आता है that is how the pollen tube is going to enter the embryo sac. पहले जो था that was entry of pollen tube into the ovule. Now we are studying about entry of pollen tube into the embryo sac. अब एम्ब्रियो सैक के अंदर एंट्री के लिए ओनली वन पाथवे इज डिसाइडेड, दैट इज फ्रॉम द माइक्रोपाइलर एंड. अब माइक्रोपाइलर एंड से ही एंट्री होगी बिकॉज एट दैट पोजिशन देयर इज द डीजेनरेटिंग सिनर्जिड सेल. So I hope in the previous video you might remember कि degenerating synergid cells जो होते हैं they are going to show filiform apparatus, and this filiform apparatus at the micropilar end is going to guide the entry of the pollen tube. So there, because of this entry, only one entry is possible from the micropilar end. So once all this entry is occurring inside the pollen, so pollen tube के जो सारे contacts है, once it reaches the ovule, then all those process is going to be called as pollen-pistil interaction. A pollen-pistil interaction is going to occur in a very systematic and all coordinated manner, that is called as dynamic process. Now let's understand some points, that is in vitro pollen germination. Now pollen germination, that is formation of pollen tube inside the stigma. अब वो तो इतना microscopic होता है that it becomes difficult to directly notice it on the plant. So what can you do for your studies? You can see the pollen germination in vitro conditions. So how can you study it? Once you dust the pollen grains, that is you can use these plants pollen grains if you get, and you dust it onto a glass slide. Now once you place it on the glass slide, you make a solution wherein you would have 10% sugar solution, and in that solution you would add boric acid, some ions like calcium, magnesium and some potassium salts. तो ये सब add करने के बाद if you pour one drop of this solution prepared by you onto the pollen grains and you leave it for 15 to 30 minutes. Now once this is settled out, what is going to happen? The pollen tubes are going to be formed. Now once you place this glass slide under the microscope, you would be able to understand the pollen tube germination. So that would be called as a hanging drop method, wherein you are understanding the pollen germination in vitro conditions. Now यहाँ पे why did we add that solution? Because that is similar chemical constituents that is happening inside the style. So style में similar conditions की वजह से the pollen tube is going to be formed. That is the reason for in vitro pollination, in vitro pollen germination, you are going to use this solution. अब एक important point पढ़ना है, that is artificial hybridization. Artificial hybridization क्या होता है? Wherein the human is going to decide that who is going to be the male and who is going to be the female parent. So this process is generally used in crop improvement processes, wherein you are going to use your superior plant varieties to make one more additional superior variety, combining both the characteristics of male and the female parent. So this process involves 5 steps, that is, first of all, you have to select the parents. Once the parents are selected, you are going to segregate who is going to be the female and who is going to be the male parent. अब जब डिसाइड कर लिया है कि मेल कौन है और फीमेल कौन है, Now you have to see whether the female plant is having bisexual flowers or if it is having unisexual flowers. अगर bisexual flowers है तो what is going to be needed next? That is, you have to undergo a process that is called as emasculation. Emasculation क्या होता है? That you are going to remove the anthers from the female parent buds before the anther dehisces. Now, why this process is important? Because female parent that has been chosen by you should not have anthers, क्योंकि अगर anthers उसमें रह जाएंगे then on maturation there is a high chance of self pollination, और self pollination की वजह से the female parent can get fertilized from the same anthers. So for that to avoid, you are going to remove the anthers, and removal of anthers from the female parent buds will be called as emasculation. Now once the emasculation process has been done by you, next step is going to be bagging. बैगिंग यानी क्या होता है कि जो फ्लावर्स के एंथर्स कट किए थे that has to be covered by a suitable sized bag. Now bag can either be made up of butter paper or it can be made up of plastic bag. तो उससे क्या होगा, बैग से अगर प्रोटेक्शन दे दिया फॉर द इमेस्कुलेटेड फीमेल फ्लावर, तो बाहर से जो पोलन ग्रेन्स आ रहे हैं बिकॉज ऑफ सम अदर पॉलिनेटिंग एजेंट्स, then unwanted pollen grains का entry would be stopped. So once it is bagged, then it is secured कि बाहर से कोई pollen grain is not going to fertilize the female parent. Now once you have done this process on the female flower, now you have to dust the pollen grains from the male parent plant that you had decided. तो वहाँ से you take some small paintbrush, collect the pollen grains, and then what you would do, you would place these pollen grains collected onto the female plant that you had selected. The female plant पे you will place those pollen grains, let them germinate, and again you will rebag it. Rebag करने से क्या होगा, they will be secured in their own isolated area and other foreign pollen grains would not contaminate your experiment. So once these are rebagged, then the process of fertilization and post fertilization events will occur. उससे fruits बनेंगे, and when these fruits are formed, that is the plant that you needed that can be grown after this artificial hybridization. अभी कंडीशन आती है कि अगर फीमेल प्लांट आपने जो चूज किया था, इफ इट डजंट हैव बाइसेक्सुअल फ्लावर्स एंड इंस्टेड ऑफ दैट इफ इट हैज यूनिसेक्सुअल फ्लावर्स, तो क्या करेंगे? सो इन दैट कंडीशन यू नीड नॉट अंडरगो इमेस्कुलेशन स्टेट, यानी कटिंग ऑफ एंथर्स आप करोगे नहीं, बिकॉज द फ्लावर इज गोइंग टू बी यूनिसेक्सुअल, सो ओनली फीमेल पार्ट ऑफ द फ्लावर इज गोइंग टू बी विजिबल, सो देयर इज नो नीड फॉर इमेस्कुलेशन. Emasculation होगा only for the bisexual female flowers. Now let's understand very important process that is going to occur once the pollen grain का जो male gametes था, once it reaches the ovule, the fertilization is going to occur. So in angiosperms only this double fertilization can be seen. So once the male gametes have been entered onto the synergids पर जैसे ही आएंगे, there are going to be two major events occurring. पहला इवेंट होगा that is called as syngamy. Syngamy यानी एक मेल गैमेट जो आएगा, that is haploid state, is going to fuse with the egg cell that is again haploid and form a zygote that is diploid. So ये पूरे process को, that is formation of zygote from one male gamete and one female gamete, that is syngamy. Second major process होता है that is triple fusion. Now why is it called triple fusion? Because it is going to involve three nuclei. Now because of three nuclei involved in this process, that will be called as triple fusion. Now which are the three nuclei? पहला होता है one male gamete that was left out after syngamy. तो ये male gamete is going to fuse with the two fused polar nuclei. तो polar nuclei कहाँ पे थे? Inside the central cell. So central cell के साथ अगर male gamete fuse हो जाता है, that is going to lead to formation of PEN. अब PEN क्या होता है? That is primary endosperm nucleus. Now this primary endosperm nucleus is going to be triploid state, क्योंकि male gamete था haploid और polar nuclei था diploid. So after the fusion, that is going to be a triploid nucleus. Now these two processes, that was syngamy and triple fusion, together they would be called as double fertilization. So here in the adjacent diagram you can understand the position of primary endosperm nucleus, that is triploid, and the zygote, that is diploid. So this double fertilization is going to be a unique feature for angiosperms, because angiosperms are only the plants those who are going to show this double fertilization process. So with this we have completed the formation of zygote. Now in the next video we will see the development of zygote and development in the primary endosperm nucleus. So till then you have to like, share and subscribe, and do hit the bell icon so that you get notified to see us in the next part of this chapter. Till then, keep studying, keep smiling. Thank you.